Welcome to the Study Explainer on cell biology. If you're looking to absolutely crush Chapter 2 of your textbook, you are in the exact right place. Today, we're not just going to sit here and read about cells. No way. We are treating this as a practical, step-by-step how-to guide for understanding the fundamental building blocks of life. I'm going to walk you through the core concepts, the essential tools you'll need, and the exact lab practicals you've got to know for your exams. So let's get that energy up, grab your notes, and let's tackle this together. Okay, let's lay out our roadmap for today. First, we'll learn how to see a cell, then understanding osmosis and cell walls, followed by prokaryotes versus eukaryotes, the organelles inside a cell, and finally, how normal cells divide. All right, let's jump right into Section 1, how to see a cell. We've got to start with the most essential tool for any aspiring biologist. I mean, you can't study what you can't see, right? So you'll need to know these microscope parts intimately for your practicals. Before we dive into the microscopic world, you have to master the light microscope. You've got the eyepiece up top where you look in, the objective lens hovering just over your specimen to actually magnify it, and the stage where your glass slide rests. And here's the trick. Knowing how to tweak those coarse and fine adjustment knobs is exactly what takes a blurry, confusing blob and turns it into a perfectly sharp, beautiful cell. Now you might be wondering, how do we actually measure something that tiny? Well, here is your exact step-by-step playbook for tackling Activity 2.1 in your textbook to estimate the actual size of a single cell. Step 1, you just place a transparent millimeter ruler right on the stage. Step 2, measure the diameter of the circular field of view you see through the lens. Step 3, carefully swap out that ruler for your prepared onion peel slide. Step 4, count how many cells fit straight across that same diameter line. And finally, Step 5, simply divide your total diameter by the number of cells you just counted. Boom! That simple math gives you the estimated size of one single cell. And hey, lock this crucial conversion factor into your memory for your exams right now. 1 millimeter equals 1,000 micrometers. Since cells are literally microscopic, you'll constantly be converting those millimeter ruler measurements into micrometers. So 1,000. Don't forget it. With our observation skills locked down, let's move on to Section 2, understanding osmosis and cell walls. To truly get this, we're going to have to get our hands a little messy. Let's walk through Activity 2.2 step by step. This is the classic potato experiment. First up, carefully cut a potato into 2 roughly equal pieces and weigh them on a balance. Make sure you record that initial weight. Second, drop piece A into a beaker of plain, normal water. Third, place piece B into a 20% salt or sugar solution. Fourth, just step away for an hour. Let the biology do its thing, and then come back to measure the final weight of each piece. So here is the kicker. What actually happened? In beaker A, the potato in plain water actually gains weight and swells up. But over in beaker B, the potato sitting in the salt solution loses weight and shrinks. Why did they do completely different things? Well, this brilliantly illustrates the whole concept of osmosis. Osmosis is just the movement of water from an area of higher water concentration to an area of lower water concentration through a selectively permeable membrane. Think of it like this. An isotonic solution means water moves in and out equally. Everything's balanced. A hypotonic solution, like our plain water beaker, means water rushes into the cell, puffing it up. And a hypertonic solution, like our salty beaker, means water is literally sucked out of the cell, causing it to shrink up. And that brings us to a massively important exam definition, plasmolysis. When a plant cell gets dropped into a highly concentrated, hypertonic solution, it loses water through osmosis. The inner contents literally shrink and peel away from the rigid outer cell wall. This is exactly why plant cells need that tough outer cell wall made of cellulose in the first place. It gives them the structural support to survive those extreme environmental stresses without just collapsing entirely. Let's keep this momentum going. Section 3, prokaryotes versus eukaryotes. Let's see how we classify cells based on how complex they are. Think of this comparison as your ultimate high-yield cheat sheet. Seriously, you might want to take a screenshot of this. It perfectly outlines the core differences between simple prokaryotic cells, like bacteria, and complex eukaryotic cells, like the ones making up your own body. Notice that prokaryotes are tiny, usually just 1 to 10 micrometers, and they lack a true membrane-bound nucleus. They just have a messy, primitive nucleoid region. Eukaryotes, on the other hand, they're larger and they pack both a well-defined nucleus and all those fancy membrane-bound organelles.
Now, what's really cool here is how we can break this down even further. We can split eukaryotic cells into plant and animal cells and compare them straight up with bacterial cells. Pay super close attention to who gets a cell wall. Bacteria and plant cells both have them for extra protection and structure. But animal cells, like ours, absolutely do not. And that's a good thing because it gives our tissues the flexibility we need to actually move around.
All right, let's crack open the cell in section 4, the organelles inside a cell. We're going to do a rapid-fire review of the tiny factories working inside the cytoplasm. First up is the absolute boss of the whole operation, the nucleus. It's a double-layered safe house. It holds your entire genetic blueprint in the form of DNA, which gets bundled up into rod-shaped chromosomes when the cell is getting ready to divide. Literally every single function and reproductive step your cell takes is dictated by the genes locked right in here.
Next up, we've got the endoplasmic reticulum, or ER for short. This is our main manufacturing factory, and it comes in 2 flavors. You've got the rough ER, which looks all bumpy under a microscope because it's covered in ribosomes that churn out proteins. Then you've got the smooth ER, which has no ribosomes and focuses on making fats and lipids. This whole factory works hand in hand with the Golgi apparatus, which is basically the cell's post office, packaging up those manufactured goods and shipping them out to wherever they're needed. Now, pause for just a second. I want you to think about this classic textbook question. A bustling manufacturing city generates a ton of garbage, right? So why are lysosomes often referred to as the cell's cleanup system? Yep, you guessed it. They act as the garbage disposals of the cell. Lysosomes are these tough little membrane-bound sacs filled with incredibly powerful digestive enzymes. They use those enzymes to break down waste, chew up damaged, worn-out organelles, and even destroy foreign invaders like bacteria that manage to sneak into the cell. They keep the whole cellular city clean and safe. And of course, no city runs without power. That brings us to our power plants. Mitochondria generate ATP energy for almost all cellular activities through cellular respiration. They are literally the powerhouse of the cell. Then you have chloroplasts, a type of plastid found only in plant cells. They use green chlorophyll to grab sunlight and synthesize food for the plant. And here is a crazy fact for you. Both of these organelles have their own unique DNA and ribosomes. It suggests they have a really wild, independent evolutionary history. Finally, we've made it to section 5, how normal cells divide. Let's look at the ultimate magic trick of biology. How does one single cell become the trillions of cells you need to grow and heal? Well, in your Activity 2.5 lab, you grow onion roots in water. If you carefully cut, stain, and squash that growing root tip onto a slide, your microscope reveals these incredibly active stages of the cell cycle. Because the root tip is actively growing, the cells are rapidly multiplying. You can actually see the genetic material physically pulling apart into different structures as one parent cell splits to become entirely new cells. It is amazing to see live. You've absolutely got to nail this distinction for your test. It's super simple, though. Mitosis is for general growth and tissue repair. It takes one cell and produces 2 identical daughter cells with the exact same number of chromosomes. Meiosis, on the other hand, is strictly for sexual reproduction. It takes 2 rounds of division to produce 4 cells, and each of those has only half the normal number of chromosomes, just like in sperm and egg cells. As you finish up your exam prep and we wrap up this explainer, I want to leave you with something truly mind-blowing. Back in 2010, scientists actually removed the DNA from a bacterium and replaced it with synthetic DNA that they created on a computer. And guess what? The cell booted up and started dividing perfectly, all based on that artificial code. If scientists like Craig Venter can already manipulate cell instructions like that, what happens when we can build a complex cell entirely from scratch using non-living chemicals? Just let that sink in as you continue your biology journey. Good luck with your studies, Class 9. You're gonna do great. Keep exploring.